Good morning, Veenu.
Hello. Good morning, Arun. Can you hear me?
Yeah, we are audible. You are audible.
Yeah, thanks. Hi, Arun. Can I share my screen now? Or we need to wait?
Yes, yes.
Okay.
So, Veenu, should we start?
Yeah, Arun. Yeah. Yeah, I'm just sharing my screen.
Hello,
Yeah.
everyone. Good morning as per Indian time. And myself, Dr. Arun Kumar Misra, Head Pharmacy Academy and Dean Student Welfare at IFTM University, Muradabad. Today is the second day of the International E-Faculty Development Program on Emerging Trends in Pharma Sector Global Perspective being organized by Faculty of Pharmacy, supported by Association of Pharmaceutical Teachers of India, formerly called as APTI. From the bottom of my heart, first of all, I would like to welcome Professor M.P. Pandey, Vice Chancellor, IFTM University, Professor Sanjeev Agrawal, Registrar, IFTM University, Professor Navneet Verma, Dean, Faculty of Pharmacy, Professor Sushil Kumar, Director, School of Pharmaceutical Sciences, and eminent speaker of today's session, Dr. Veenu Bala, Clinical Pharmacology Laboratory, Department of Pharmacy Practice and Sciences, University of Nebraska Medical Center, Omaha, USA, and all the faculty members and my dear students. From my side, it is welcome of all the members associated at here. Now, I would like to give a brief introduction of our eminent speaker, Dr. Veenu. Dr. Veenu is associated with the Clinical Pharmacology Laboratory at University of Nebraska Medical Center. She did her B Pharm from Rajasthan University in 2006 and M Pharm in Pharmaceutical Chemistry from IFTM in 2008. She did PhD from CSIR CDRI and having 7-year plus experience as pharmaceutical professional. For her PhD thesis, she was awarded with Dr. M.M. Dhar Memorial Distinguished Career Achievement Award 2015 for Best PhD Thesis from CDRI, Lucknow. She worked as Assistant Professor at Mohanlal Sukhadia University, Government of Rajasthan, Udaipur, between 2014 to 2018. During that tenure, she was recipient of UGC Startup Research Grant for university faculty from November 2015 to October 2017. At University of Nebraska Medical Center, USA, she earned expertise skill on population pharmacokinetic modeling while working in analytical clinical pharmacology lab. She is certified population pharmacometrician and NCA analyst from Certara using Phoenix software. She has to her credit 2 patents and 35 international publications of good repute. These are some of her achievements and accomplishments. In this short span of time, it is impossible to share her achievements more. So now I would like to invite Dr. Veenu to deliver her talk on clinical pharmacology research and career perspective. Veenu, it's over to you.
Thank you so much. Thank you so much, Arun. I really appreciate the nice introduction. Today I'm going to talk about like how clinical pharmacology is involved in the drug development process. Let me start with my presentation. You can see here when we are looking at the paradigm of drug development process. Drug development or drug discovery pathway, as you can say, it starts from the bench study. Then we are moving towards the non-clinical study, and after IND approval, mean investigational drug approval from FDA, we are entering in the clinical study, means the study which is done inside the human body. And after these clinical trials, we again need FDA approval, and we file the NDA towards our FDA approval, and then we move towards the phase 4 clinical trial study. So the whole study which is done in the human, like those clinical trial, phase 1, 2, and 3, it is often understood like only phase 1 clinical trial study is related to clinical pharmacology research, but it is really not like that. The clinical pharmacology research is usually goes within or by side of all these phase clinical trial study 1, 2, 3, or either 4. It is a kind of post-surveillance study. Some clinical pharmacology data is also collected while we are in the phase 4 clinical trial. Because of when the drug is in the market, like you guys are also doing research, I think, on nano formulation, different dendrimers formulation, or different dosing kind of or formulating different kind of dosing strategies. So those things come under the phase 4 clinical trial. So it is also kind of clinical trial study. So in all the phase 1, 2, and 3 clinical trial study, we are collecting the data and the what actually we want to do with our data. If I'm talking about in terms of FDA really wants data in its own terminology. It is clearly defined in FDA guideline what actually it wants in every phase 1, 2, 3. As I said, the major studies of clinical pharmacology is done under the phase 1 clinical trial study. Like we are checking it is a checkpoint for the safety analysis, but it is done in the healthy human being. And then phase 2 study is a checking point for the efficacy study, and efficacy is really done in the diseased patient population, and phase 3 is kind of confirmatory test for those efficacy study, those safety study. So all data is collected in all those 3 or 4 clinical trial study, and all those phases comes under the clinical pharmacology research. And as I told, like safety, efficacy, and results are collected here. Like FDA is having a clear-cut guideline or expectation, I can say, that what what exactly FDA want in every clinical trial study. So if I'm going towards the clinical pharmacology definition, or I want to define properly clinical pharmacology definition, like it is a kind of making use of clinical data that allows a physician for the proper use of medication. And why a physician want proper use of medication? Because he want to achieve a optimal effectiveness, and also he want to manage side effect, like least kind of side effect a physician want while he is prescribing a drug. So in the whole clinical pharmacology, we are designing the clinical trials over here. Like first, we want to done the clinical trial in the healthy human being for the safety perspective, and then we are moving towards the diseased population to find out the efficacy of that data, not efficacy like a end point of decision making. And the whole data, whether it is in the healthy human population or whether it is conducted or collected in the diseased human population, it overly based upon, there is a need to point out that this data, whether it is scientific or analytical data, it is a key point in a decision making. So in a layman language, like we are saying that drug effectiveness, we are working towards a drug effectiveness, like in terms of, if I'm talking about pain reducer medicine, what a layman will understand, he wants that the pain is reducing. If a person is having fever, the fever is gone, it's done. And if I'm talking about sleep medicine, if I am able to get sleep after taking that medicine, the work is done. But it is not like that. The effectiveness is not like that. Clinical pharmacology thinks something extra than this. In clinical pharmacology, we are basically dealing with the PK and PD profiling of a drug. As the name suggests, PK pharmacokinetic, PD is pharmacodynamic. In PK, uh, I think everybody knows that definition, what the body does to the drug. It is related with the absorption, distribution, metabolism, and excretion of a particular drug candidate. And PD is the what a drug does to the body, means how it is showing its effectiveness within the body side. And along with these, on clinical pharmacology is not only the study of these two, PK and PD. Along with this, there are several studies are conducted, or several different kind of clinical trials are conducted, in which we are assessing the influence of physicochemical property of drug and also the patient's intrinsic and extrinsic factors over the PK and PD profile of that drug. Like physicochemical properties, you can say like different formulation, different product, different administration route, and patient's intrinsic and extrinsic factor, like intrinsic factors like its organ dysfunction, renal failure, renal dysfunction, its hepatic dysfunction, different disease, and extrinsic factor like concomitant medication, different co-administered medication effect, food effect, or you can say its own parameter effect, like its gender effect, and also like its own habit effect, like whether the person is a smoker or non-smoker, drinker, non-drinker. So those all things are taken into account while we are conducting any clinical pharmacology research. Along with these PK and PD study, there are several other pharmacogenomic studies are carried out, several toxicological studies are carried out, and also the dosing schedule or dosing regimens are also decided. Like, some drugs we are taking one tablet, some person is taking two tablet if it is not sufficient, some are taking 500 mg dose. Like in PCM, you can see there are two different doses are available. I think one is 500 mg and one is 650 mg doses. So those things are decided while we are doing the clinical pharmacology study, whether we have to take the drug as a OID dosing once in a day or BID dosing. So those all things are decided while we are conducting the clinical pharmacology research. So what exactly we want from this whole clinical pharmacology study? But the ultimate goal of this clinical study is like we want that the right drug should reach to the right patient, and not only reach to the right patient, but the right dose and right timing should be there. When all those four goals will meet, then it will be really effective a drug therapy for what purpose it is sought for. So it is the ultimately goal of a clinical pharmacology research. So when we are having the answer that what exactly it is doing, we have to know that how we can achieve that goal. So I'm giving you idea like how different kind of clinical pharmacology studies are conducted, what kind of studies are conducted here. The very first study, when we are moving from the non-clinical to clinical species, it is the first in human study. So like I am doing experiment in a monkey species, I want to do in a human study then the first problem which came, or first aspect which I have to look is how much dose I have to give to that human. I can't dose that human like a monkey dose or like a mouse dose. So there are several parameters, several non-clinical studies. So actually we have to look towards the non-clinical study, and we have to find out, and there are several studies we have already carried out in non-clinical part. ADME study is also a part of non-clinical study. By those study results, we have to decide at what dose we have to start our phase 1 healthy human clinical trial. And the thing is not lasting only with the only one dosing study. After that, there are multiple single ascending dosing study, and single ascending dose study and multiple ascending dose study. PK studies are carried out. Single ascending dose study, we are taking one dose, only one dose a day. We are dosing those human being as a once a daily dose, but we are increasing the amount of that dose, like first 500 mg concentration dosing study, then 7, then 800. And because we want to assess the safety profile, how much time we are not going inside the toxic level. And then likewise, that multiple ascending dose study are carried out. And after that, healthy and patient population, we have to carry out different study with phase 1 is healthy people study, and phase 2 study is a patient population study. Phase 3 study is also there. And like we have already done ADME in non-clinical species, but we have to done in a clinical species. So along with these study, we are taking the sampling, we are collecting the sample, and we are assessing its ADME properties. And as we are moving forward in the clinical pharmacology study, apart from only these dosing study, we have to taken into account the specific population. Because when we are using that a medication, what we really want a single dose or single kind of medication for a whole population, where like everyone can take. But the dosing should be decided over there. It typically depends upon how the patient is behaving. Like I'm giving a drug to a renal impaired patient, so how much dose I have to give to that patient. Like one of my drug is cleared from the urine, like it is excreted 90 to 100% excreted through the kidneys. So these things should be taken into account. Like if a person's kidney is not working fine, so I can't dose him exactly the same dose as I'm giving to the healthy human being, because otherwise it may cause toxicity. It is not excreted. Like if the dose is given BID, like twice a day, I'm giving the similar dose to healthy human and similar dose to renal impaired patient. The renal impaired patient will develop toxicity. So those things or those studies are also carried out. Mainly renal impaired and hepatic impaired studies are over there. And after that, in this situation, now we are in the situation like a risk assessment kind of situation is there in clinical trials. We have to assess also age factor. We have to carry out the study in different aging group. Like 20-year people we are taking, and we have to take into account the 80-year older person we have to also taken into account. And then we have to take into account the gender studies are also there. Like when we are starting to the clinical trial, the very first clinical trial is usually conducted in healthy male, but it is not always there, because some of the medicines or some of the drugs are particularly sought for a female use. So that time those scenario change. And then after we really want to do those study in a pediatric population also, because it is usually not like that. Is a healthy male is taking a full tablet, we can give half tablet to a pediatric patient. So we have to done the study over time, because it's fine for a pain reducing medicine, but it is not fine when we are talking about a cancer drug or CVS drug. So those kind of studies should be taken into account. And after that, when we are moving forward, like there are several drug interaction studies when a person is having any chronic disease, already a chronic disease like CVS disease or CNS disease, and then we have to give him another medication. So how that medication will interact with each other. Like we are already giving drug A to that person, and now we want to give drug B to that person. So we really don't want to any kind of antagonistic or synergistic effect. We really don't want to cancel out the effect of drug A when we want to give him drug B. And there are one really popular nowadays aspect in clinical pharmacology is population PK. I'm basically working on that aspect here. of population PK, in which we are collecting different phase clinical trial data, and we are making some mathematical algorithm setup, and we really want to narrow down the requirement of those clinical pharmacology study. We want to narrow down, like, we don't want to do this kind of several kind of study, because it's not always feasible to find out those human volunteers are there. If I want to do a pediatric kind of population, it's not always feasible, like I get a complete enrollment of pediatric patient for that particular drug. And if I want to carry out a study in a pregnant woman, it's not feasible, like we got that kind of patient population. So those are the main clinical pharmacology study which is really required while we are moving towards the NDA filing. And after that, there are several exposure response studies are there. These PKPD studies are mainly the part of phase 2 and 3 trial study. Also the biopharmaceutical study, bioavailability study, bioequivalence study, when we are talking about different formulation. One more really very nice study is usually done in clinical trial is the food effect study. Its studies is done in the presence of food or absence of food, whether that drug has to taken early morning, whether it is taken with food or whether it is taken after food. So those study, and also some kind of in vitro studies are also done. These are mainly done along with the non-clinical study because these are in vitro study, and we are using the human microsomes, hepatic enzymes for conducting these study. And for those studies, several bioanalytical methods are developed, and some kind of biologics only study when our medicament or our drug is related to biological compound. So immunogenicity and comparability studies are over there. But basic studies are of this side. Those clinical pharmacology studies are mainly done while we are in the phase 1 clinical trial. One thing I really forgot over here, the racial difference study is also done over here. If you want, like, our medicine should be used globally, I want to use my medicine like not only in India, I want to use in America, I want to use in European culture, because in European, the Caucasian population is over there. So they are having some metabolizing enzyme difference within those population. Some population like Japanese population are poor metabolizer of CYP3A4, and those Caucasian population are having different metabolizing effects. So those things or those studies are also taken into consideration while we are conducting clinical pharmacology research. This is a really it is a FDA paradigm. It is taken from the FDA, which is clearly indicating the timing and early clinical pharmacology study. Like it really divided what kind of study is taken in which phase. These are the preclinical study, our assay development and validation, which we usually do via the LCMS-MS, and our single ascending and multiple ascending dose study, which is the main study of phase 1 clinical trial. PKPD is part, but PKPD we are assessing, but how we are assessing, we are using single ascending dose study, multiple ascending dose study, and then we find out the PK and PD, how it is affecting our PK and PD. And food effect study is also part of this study. Then phase 2 and 3 study, basically their PK and PD study is done in the patient population. And here you can see like PK in renal impairment and hepatic impairment studies carried out. And one thing should be always clear, like while we are going towards the NDA filing, there is no room or no chance that we can submit any drug to NDA. We can do any NDA submission to FDA without conducting a renal impairment and hepatic impairment study. So it is a must required study for NDA filing. So ultimately, what we got after conducting so many clinical pharmacology research study, if you remember, like when we are getting any medicine from market, if it is in a carton, we are having a leaflet inside that carton. So the whole leaflet information is come from that clinical pharmacology research study. It is having information about indication, uses, doses, administration, like this complete list. It is having all those study in over that leaflet. And we are not reading that leaflet, but actually physician is reading that leaflet, because he has to make a decision how he can prescribe the medicine to the patient, what kind of dose he can prescribe, what kind of schedule he can prescribe to a diseased or to be effective in a particular disease. So those clinical pharmacology research, it is a kind of impact of those research, like these clinical pharmacology research study mainly impact the labeling of that medicine, which ultimately affect the dosing schedule of that medicine. I'm just here showing some PK/PD graphics to show how these clinical pharmacology PK/PD studies are done. What kind of graphics usually come in forward to us. We all know that in PK study, it is a concentration time profile. We are really checking the exposure of our drug, how much drug is reaching inside our body, what is the threshold, how much threshold it is reaching over there. And in the PD study, what we are finding out, like how much that concentration is giving us the effect. So if we combine those PKPD study as we are combining in the phase 2 and phase 3 clinical trial study, it is a kind of exposure response study. uh, Here you can directly see that as soon as the exposure or concentration in the blood is increasing, our response is also increasing, and there is also a plateau phase. After that, there is no other response increase in, there is no increase in response. It is like a not a direct response exposure relationship. It is, you can say, it is sigmoid shape or indirect response exposure relationship. But it is not always like that. You are increasing your doses or increasing your drug or medicament, like I'm taking 100 mg drug, I'm increasing it 200, I'm increasing it 300, 400, and my response is linearly increased with exposure. So it is not like that. If we are increasing the dose after a threshold, it always converted into a toxic effect. So those things should be taken into account while we are conducting any PK and PD study. And those clinical trial PK PD studies give us idea that how much drug should be taken, like how much Cmax we have to decide. There is a kind of therapeutic window. Our doses should be adjusted like that. Our effect or response or our exposure should be in between that therapeutic window. So these PKPD studies are really a backbone of our clinical pharmacology research. Without those PKPD study, like drug is nothing. It will never move forward without those PKPD studies. Here I'm also just representing that concentration time profile. It is a PK profile. You can see I just want to show like how this concentration time profile is changing with respect to diseased patient, like patient with hepatic impairment. They are hepatic impairment, and I know that my drug is mainly metabolized by a specific hepatic enzyme. So when a hepatic impaired patient is taking that drug, and it is in the same concentration as the healthy or different the diseased population, then the Cmax is increased, and this increase in Cmax is not very good for the safety purpose. So the drug will not move forward in the phase 2 clinical trial. And it is also true in different genetic rapid metabolizer, like their hepatic enzymes are very fast, or they are having they are known as the rapid metabolizer. It depends upon different races. So they metabolize our drug very fast, and it is unable to reach the Cmax. So the dose we are giving them for a particular exposure, it will not show any it will not show our desired effect. So it will be ineffective in those those rapid metabolizers. So the dosing should be adjusted like that. It should at least reach the Cmax. Here I'm giving some example of different clinical trial study, like a food effect study is carried out for atazanavir. Atazanavir is a antiviral agent which is really used in anti-HIV combination therapy along with the efavirenz. So for its clinical trial was conducted under the fed condition and under the like mainly high fatty meal affected this compound. So the clinical trial was conducted in two subgroup. The population was divided into two subgroup in which the same dosing was given to one population which is having the light meal like non-fat meal, and the other population which is already fed with a high fat meal. So they find out that the Cmax and AUC difference was there. Cmax for light meal patient was 57% and high fat meal, the Cmax was actually not changed, but AUC was changed because it was affecting the clearance of that compound. AUC, when we are taking the light meal, it was 70%. When it is given a high fat meal, the AUC was increased by 35%. So those things were taken into account, and the clinical trial were conducted under fed condition, and the empty stomach or light meal direction, and it really gave a really like actual direction which was included in the label, that these drugs should be administered with a meal or snack, not high fat diet but with a light snack and meal. There is one more example of renal impairment study with the doripenem powder. It is a radio-labeled ADME study. The drug is really very much excreted in the urine by the 12 hours. It is more than 90% excreted in the urine, and as the primary elimination site is kidney, so it should be taken into consideration. Like renal impairment study was conducted, and a full PK study in patient with renal impairment were conducted, and phase 2 and 3 trial doses was adjusted, which was based upon creatinine clearance, and label correctly or recommendedly indicate that its dose should be reduced in patient with moderate to severe renal impairment, and hemodialysis as a treatment of overdose. If it is overdose, if you can't reduce the dose less than a certain amount, and the patient is highly renally impaired, then hemodialysis should be done over there. So as I give this much example, like how clinical pharmacology studies are carried out, so you can really see over here where we started. The base of clinical pharmacology is a very thick base, and after when we start going upward, it is getting narrower, but that narrow things, like we are as a resultant, we are getting like as a resultant of all those study, like how the dosing and our dosing schedules should be adjusted. But it is the main basic of any medication, like how much drug should be given. We are making the drug, it's not like that. How we are using that drug, how the physician is prescribing that drug, that is ultimately the main goal of any research, not only clinical pharmacology, any bench research or any non-clinical research, clinical research. But actually we are starting with the clinical pharmacology study, and we have our narrow paradigm decided that how much drug should be dosed and administered to a patient population. So here the question comes, why we need these clinical pharmacologists? If I'm talking about American market, as the data is showing, like about 50% of American take at least one prescribed medicine, and more than 23% take 3 or more prescribed medicine. So like this much medicines are formed, or this much medications are formed over here or consumed over here. So it is a kind of $1 billion market. So when there is more consumption, there are more requirement of daily like newer kind of therapies, newer kind of research is going on. So there is always a need of clinical pharmacologist, and it is a kind of clinical pharmacology. It's not like that I'm a scientist, I'm doing one kind of research. It is a full-fledged kind of process. So there are different phases where a person can be recruited, where the person can work over there. So it really a huge market in which clinical pharmacologists can work over there. And if we are looking towards the cost of developing a drug, you can directly see when when we are talking about how when we are discovering or when we are studying a one drug molecule, when it come from the bench to market, it took around dollar more than $1 billion expenses over there. So there are a lot of expense, so there are a lot of demand of work power to do that kind of work. So if I took this data from FDA. If we really see the FDA registered clinical trial right now, it is more than 400,000 research studies are really right now conducting within the 220 countries. And last 2 years COVID-19 clinical trials are also going on, and currently there are more than 7,000 clinical trial on the COVID is really registered within the FDA. So to conduct those much clinical trials, to conduct those much research, there is always a demand of clinical pharmacologists at various stages of clinical pharmacology research. uh, and if I'm talking about the FDA approval rate of a drug, cancer drugs are least success rate, like only 5.1% drugs that are in the phase 1 trials ultimately reach for the FDA approval. Like only 5% drugs which entered into phase first trial move towards the phase 4 trial. And phase 2 trial is the trial where most of the drug fails. Only 31% of the drug which enter the phase 2 trial which goes to the phase 3 trial, because it is a kind of efficacy study. If the drug is having safety, but when we are try to maintain our safety profile, it is not always possible that we can maintain the efficacy profile. So that is the basic reason behind why our drugs are mainly fail in the phase 2 clinical trial. And now basically how the career is oriented in this field, clinical research or clinical pharmacology research, there are a lot of pharmaceutical companies who are paying for those clinical trials. There are one newer technique or one newer aspect of population pharmacokinetic is over there, not modeling, it's only population pharmacokinetic. It is a kind of decentralizing clinical trials. One kind of trial is conducting this one side, and other companies conducting different trial. So mainly CROs are working for those clinical trial. So it is a kind of decentralization of clinical trial. So there is also a requirement of work power. There are requirement of volunteers also. If a smaller scale we are seeing, they always need healthy human volunteers. There are also requirement of different doctors, pharmacists, clinical research managers. And also what is the whole clinical research process, what actually we are doing, we are playing with the data, we are conducting the research, we are getting the data. So there is always demand of that data managers, regulatory affair managers, because IND filing is not a small deal. It really require a work power for IND filing. And after that IND filing, I'm not saying IND filing maximum work power require, because NDA filing are not that much. The IND filing, medical writers are required, who will write their IND filing document, who will write their NDA filing document. So those things are required over there. Also the person who can monitor those clinical trials, and also those person who are conducting those clinical trials. There are always hospital, the sites where the trial will be conducted. So also the trial will be conducted, there's blood sampling will be collected. So lab investigation team should be also there, who will analyze the samples, who will take starting physical data, and those kind of requirement is always there. So what are the basic eligibility requirement if we are considering a clinical pharmacology research? It is in a clinical pharmacology research, there are chances of when all medical personnels are working very closely and collaborative manner, like different doctors, MD, nurses, pharmacists, laboratory assistants. Everybody is required to carry out those clinical trials, those clinical pharmacology research. And the basic degree start from the bachelor's degree, because you can see different stages are there where the personnel is required in this clinical research. So as I was telling you that I'm basically working with the population PK modeling study. So why those studies are considering nowadays at a high demand mainly here at USA, because they want to avoid some kind of clinical trials, like as you see last different Covaxin or those COVID vaccines are over there. If the guys start clinical trials over there one after, it was not possible that those drugs come in market within the 1 year of that infection, because there are some mathematical modeling and simulation studies are carried out by using different softwares here. So in this study, what we are doing, we are actually predicting the concentration of effective drug concentration within the body by using the different software. If we are having only single dose study in the healthy population, and as I told that in clinical pharmacology, we are going to have a single ascending dose study, multiple ascending dose study. So we are not actually conducting that clinical trial. What we are doing, we are using our mathematical software, and in that software, if I'm having the data of first clinical trial study, I can really predict the clinical trial. I can change the dosing schedule. I can change the dosing amount. I can change we can change within that software, but it is really typical, and it is in the demand of nowadays, how those modeling and simulation you can do. And we can also predict that concentration. I'm only having a healthy human patient data, and I want to in the phase 2 clinical trial, but phase 2 clinical trial, we want a lot of diseased patient subgroup, pediatric patient if we need, elderly patient, renal impaired patient, but we don't want to, we are not able to recruit those kind of patient, and it is a kind of very highly consumption market. So what we are actually doing, we are using mathematical modeling and simulation software. We are simulating that patient subgroup. We are entering that data over there, and we are just predicting the concentration level. And it is really, right now, some of the medications are having on their label that their dosing was decided by using the modeling software. So those things are also under consideration while we are developing, or while we want to avoid those clinical unnecessary clinical trial, I can say. So if I'm just defining population pharmacokinetic, what actually population pharmacokinetic is. In population pharmacokinetics, we are understanding the variability in drug concentration in different individuals or different population, and we are really taking into account different patient characteristics, which known as covariates, like different age, different disease, demographic, different gender, different concomitant medications, and if there are any disease and hepatic impairment is over there. So we study all those effects in population pharmacokinetics, and we want to understand that how that variability assessing, how we can assess that variability in that particular population, and we can establish a PK/PD profile for a effective and safe dosing regimen. So those things are done with the help of population modeling. So in this population PK study, the data is collected all over like different population subgroups. Like one person is in England, but it is the obese population, one in Indian population, healthy person, one pediatric patient, one geriatric patient. So all those data is collected overall world, and those data is combined, and we can make our model by using different clinical trial, and we can really reduce the clinical trial, actually mainly the dosing schedule studies, or ascending dose, or single ascending and multiple ascending dose study. We can really reduce that studies. And by using this population PK and exposure response modeling, we can pick a optimum dose for phase 2 and phase 3 clinical trial. If I am having phase 1 clinical trial data, there is no need to do the same ascending dose study, same single ascending or multiple ascending dose study in phase 2 and 3 clinical trial. We can find out the optimum dose by the help of our phase 1 clinical trial data, and we will just conduct the phase 2 and 3 trial study on that particular dose. So it is really a nice thing of this population PK modeling. because as I told earlier that most of the drugs fail in the phase 2 clinical trial, and if we are setting up a particular optimum dose, so there is a failure chances is really reduced. And in the population PK modeling, we can avoid clinical pharmacology study like renal impairment study. Though those study we need not to conduct at a large scale. We can just input the renal impairment profile in our software, and it will give us answer how the renal function will impact the variability in a PK and PD profile. And it will also give us information about the labeling of dose. So as I'm telling that there are several drugs are right now in the market, like which are showing that uh, that dosing profile was set by using the modeling software, not by the a usual clinical trial. It is a kind of virtual clinical trial was conducted. And one more study which we can do by these modeling software is we are conducting the in vitro study, and we can correlate those in vitro in vivo correlation study by using the clinical bioequivalence softwares. So those kind of studies or population PK modeling softwares are over there for which we can study. Here I'm just giving a link over there of this software. It is a monolith software. It is freely available for academic setup. If anybody want that software, if anybody start to use population PK study, so they can directly go to that website. They are having their own YouTube channel. You can find out a lot of videos how to do those kind of modeling over there. And also uh, and also they are also conducting some kind of spring courses, and there are one more software, Phoenix NLM is there, which basically I'm using here. So those kind of software this is really the unpaid kind of version monolith software. And the next main question, if in academic setting which came in mind, like from where we can get the clinical trial data if we want to conduct, or if we want to do this population PK study by our own self. So we really, there are a lot of data in our literature also. You can like in those software, but we can do if we are having a already generated clinical trial population PKM model, we can take that model from literature, and we can change our population subgroup, and we can change some like co-administration dosing, and those kind of studies you can really do in academics setup. So these softwares are really nice, which can be useful for a population PK study. So yeah, that was all about which I wanted to tell you about clinical pharmacology study. So I'm really thankful to you all for your kind patience for listening me. Yeah, thank you so much.
Thank you, Dr. Veenu. And now it's time for question answer. And if any one of us is having any kind of question, please ask. Dr. Veenu is with us. He will definitely answer the question.
Hello, madam. Good morning.
Good
Myself,
morning.
Rakesh Kumar Singh. Madam, can you explain in detail what is the eligibility requirement for pharmacist in clinical trials?
It is a kind of actually what I can tell you, like here in US setup, I can tell you, like here Form D is basically required for clinical setup. But in our India, I think clinical pharmacy related masters is required over there.
Okay, okay,
And it also
okay. Thank
depends
you.
upon what kind of work you are. It's not always necessary that you are directly going to conduct the clinical trial. You can be a part of any portion of that clinical trial. Mainly, like main people who are working like masters, they are working for the clinical data management kind of position in different industries who are conducting clinical trials.
Okay, okay. Thank you, madam. Thank you.
Thank you. Welcome.
Thank you. And is there anyone is more to ask? Okay, so now I wish to conclude this session, and I wish to also conclude the e-faculty development program. So in yesterday's session, we learned from Dr. Adesh that career prospects are too much lucrative for our students, and if they wish to explore their career in Australia and in other countries as well. From Dr. Adesh, we learned that various exams of Australian government, which is to be qualified for being registered pharmacist. Dr. Adesh in yesterday's session told about the academic curriculum of Australia and compared with Indian pharmacy academic curriculum. Definitely, Dr. Adesh, we will share this valuable information to our students so that they may explore the career options in other countries too. And so far as today's session is concerned, I am really thankful to Dr. Veenu. And in today's session, Dr. Veenu covered the contemporary research being carried out in clinical pharmacology with respect to pharmacokinetics and pharmacodynamics with clinical pharmacology. And she beautifully explained and discussed the various career options for budding pharmacists in a very appreciable manner. Really, this in-depth knowledge will be beneficial to us for our further studies as well. And I am fortunate enough that both of the eminent speaker, Dr. Adesh and Dr. Veenu, are my batchmates from IFTM. And from the side of IFTM University, it is also a matter of pride and prestige that my colleagues are well settled and working well beautifully in the different parts of globe. So thanks to you both from IFTM University family and especially our Vice Chancellor sir, Dr. Mahendra Pandey sir has told me to convey his blessings to you. Especially thanks to Dr. Veenu that I think as per US time in US timing it would be around 12 o'clock of night. I think so.
Yeah.
No, really, it's very appreciable that you sacrificed this much time and you shared this much valuable time and your inputs and kindly convey my thanks to Yaspal sir also because he is every time he is ready to help and definitely he helps in several kind of academic activities into us. So thank you all. And now I would like to invite our Sushil sir, Director School of Pharmaceutical Sciences, for a formal vote of thanks. Sushil sir, over to you.
Okay. Thank you, Arunji. Thank you, Dr. Veenu.
Thank you so much, sir. It's really nice to see you after a long time. Yeah.
Yes, yeah. So I am very happy to see you after a long time. Thank you, Dr. Veenu, for giving information about drug discovery and clinical trials and clinical pharmacology. We have discussed very informative information with us and with all the faculty members. So thanks a lot for you. And really, it is a very great pleasure for all of us and also IFTM family because you are working at a prestigious state in USA. And thank you so much for sharing your views with us. I am also thankful to our Honorable Vice Chancellor, Sir, Professor Mahendra Prasad Pandey, Sir, for their wishes and support to organize this faculty development programs. I am also thankful to respected our registrar, Sir, Professor Sanjeev Agrawal, Sir, for their valuable support and wishes. I am also thankful to Dr. Navneet Verma, Sir, Dean Faculty of Pharmacy, and Dr. Arun Kumar Mishra ji for their kind support during the faculty development program. I am also thankful to Dr. Harpreet Singh and other faculty members for their kind support during the faculty development program. So I am thankful to all the other staff and especially I am thankful to Professor Arun Kumar Mishra ji for their kind support to organize this faculty development programs. So over to you, Arun ji.
Thank you, sir, and thank you all. Now the session is over, so we are closing this meet. And once again, thank you all.
Thank you, Veenu.
Thank you, sir. Thank you, everyone. Thank you.